Engineering Fair. Excuse my scrolling. Don't want to make anybody sick. Okay, so tonight we're doing potentially hazardous biological agents, so PHBAs. We're going to look at hazardous chemicals, materials, or devices, and then we're going to look at display and safety, and that's pretty quick, just some things mainly if they're going to the regional fair that they need to be aware of, because we don't want them to get disqualified for putting things on their board that are not allowed there. So with PHBAs, the biggest thing in this first column, it is the responsibility of the student and all adults involved with a product or project to conduct and document a risk assessment form, which I've said everybody has to do regardless, because there's risk involved with everything we do. So the main reason is to define the potential level of harm, injury, or disease to plants, animals, humans that could occur with working with these biological agents. So our main thing really is just safety. So when we move into the rules for all of PHBAs, prior review and approval is absolutely required, because remember, with these, you're looking at bacteria, viruses, fungus, parasites, anything of that nature falls under PHBA. So you want to make sure that your students are staying safe, they're doing it in a lab. These kind of things, need to be done correctly to make sure nobody gets sick. So an affiliated fair SRC, IBC, depending on where they're doing it, has to approve everything before the research experimentation begins. And as we move down, you'll get what those acronyms stand for, the IBC and IACUC. We'll get to those in a little bit. They should have put it right there, but we'll get to what those stand for. So number 3, experimentation involving the culturing of potentially hazardous biological agents, even BSL-1 organisms, is prohibited in a home environment. So students cannot culture anything at home. They have to go to one of our high schools that is a BSL-1 lab. Obviously, if that's the only place that we have. So they may be collected at home, but they have to be immediately transported to that laboratory. So I know some students, they want to get collections from their toilet seat at home, whatever they're getting. They just have to make sure that they, water samples, anything, it's got to come to the lab once they're collected. So anything that is determined to be a BSL-1, the research has to be supervised by that designated supervisor or qualified scientist. The student has to be trained in standard microbiological practices, so that kind of thing. Gloves, alcohol, it's whatever disinfectant that person's using. It depends, on what they're using or what they're culturing, anything like that, on what that standard of microbiological practice is. And most of our high schools do have someone there that can help them with this. Those sheets have been coming in still. So as soon as I have that list of the ones that I know for a fact are BSL-1, their checklist is done, then I'm going to send that list out to everybody, so that I've already got some people inquiring because they've got junior level that want to do BSL-1. So
okay, so basically any of us running a high school lab are pretty much BSL-1, right? I mean,
yes,
if we've taken the county trainings, all the lab safety trainings and all that stuff, that qualifies us?
No. I need you to, all the high schools, I need them to do there's a checklist.
Right.
I can email it to you, or it's in that forms folder.
Right, I looked at the forms thing already there. So if we can click on all those check boxes, then we're good.
Yes. Okay. And that's why I say you really just need somebody there that does know that if they've got something that's being cultured, that person needs to be in the room. You need to make sure that those culture dishes or anything growing in them are bleached and disposed of properly, or autoclaved if you have an autoclave. So those kind of things, you do have options there. Just you need to make sure most high school labs have all the equipment you need to make sure that happens.
Right. So basically we could sign off on that.
Yes. Well,
in our labs. So
yes, you go through the checklist, and then you need an administrator to sign it, preferably your principal.
Right. Okay. That's what I thought. I figured we have all that stuff in our labs, so I'm figuring I don't, it says disinfectant or autoclave, and I'm like, well, we have disinfectant. We supposedly have an autoclave somewhere, but definitely have the disinfectant. So
exactly. So
Okay.
yeah, you may have somebody, a student from Bellevue Middle that wants to come over and do a BSL-1 lab there.
Hmm.
So, and I just want to make sure our junior level students have that option, because we do have some that are on that kind of intellectual level and they want to be able to do that kind of thing. Like I said, I've already had a few people email and say, hey, when are we getting that list? I've got some wanting to go to the high schools and do a BSL-1, which is really exciting because at junior level they could really increase that competition for them to be able to go to state.
Okay.
So that would be really cool. All right, any other questions?
Nope.
Move into BSL-2. All right, so any kind of BSL-2, that is going to be at a regulated research institute, period. We do not have anything in a high school lab that, I've looked at the checklist. We don't have that kind of things in our classrooms or accessible to us. So those have to be done at another institute. So here's IBC, that Institutional Biosafety Committee. So they would take that checklist there with them. Donna just emailed, can you let DMS know that they can use us? Yes, ma'am. What I'm going to do is once I get everybody's biosafety level checklist, I'm going to email that out and say, hello, hello everybody. If you've got junior levels that are wanting to do a BSL-1, go to the high school that they would go to if once they leave middle school. So DMS would definitely go to Denellen High, Bellevue Middle to Bellevue High. So those kind of things, go to the closest high school to you that is a BSL-1 lab. So I know, yep, I will make sure and get that list out. I'm still waiting on a few more papers to get here. All right, BSL-3 or, oh, DMS is Denellen Middle School. Sorry, just had that question. Who is DMS? Okay, number 6, prohibited. We cannot do BSL-3 or BSL-4 research. Now I know some of you may be going, what the heck are all these BSL-1s and 2s and 3s? Once we get through this part, there's actually an entire page that defines those and what they look like and what kind of things can be done and gives you some examples. So we will get to that point shortly so that those kind of confusions can be cleared up. Because I know it's a lot of acronyms. The science fairs is acronym happy as education in general. So any lab studies that they're going to culture known clinically significant multi-drug resistant organisms, so MEDROS. So here we go again. They must have written justification for usage and be conducted at a regulated research institute. So anything that is like resistant to antibiotics that could potentially make students sick if they were to be contaminated with it, they've got to be in a BSL-2, and that's why. Miss Crusher, you can't hear me at all, or you can't speak to me. I'm going to give her a minute to answer that. Okay, so some examples of things that are in that resistant organism group. You're looking at MRSA, which obviously we know we don't want our children playing with. Lindsay Barber's down here giggling at me while I do this. So it's got some listed right there, the VRE, CRE. Most of our students hopefully aren't, in here playing with these kind of things anyway, but just those organisms that obviously are going to make us, really sick. And then any kind of yeast or molds with a known resistance to an antifungal agent. Okay. So some of this gets a little crazy right here. Students may not genetically engineer organisms with multiple drug resistant traits or intentionally select those organisms through passage and culture with the purpose of investigating pathology. They don't need to be developing or treatment of antibiotic resistance infections. So pretty much stay away from that. Hang on, trying to help Miss Crusher out. She can't hear me at all. We'll see if that helps. Okay, so basically anything that's antibiotic resistant, they've got to stay away from, or it has to be in a BSL-2 containment lab because of obviously the just the risk of infection would be high if it's antibiotic resistant and they're playing around with it. This one's all kinds of fun. Culturing of human or animal waste, including sewage, sludge, they have to be in a BSL-2 lab. I'm not sure what excited student's going to do that, but they could definitely find some interesting things. But just remember, if they choose to do that, it has to be in a BSL-2. So that means they're going to have to go to a research institute like UF or SHANS or anything like that if they wanted to culture those kinds of things. Hang on just one second. All right, I'm back. Sorry, Mr. McAteer had a question. All right, where we were. All right. The PHBAs have to be properly disposed of at the end of experimentation in accordance with their BSL level. So remember, your BSL 1s, they can be autoclaved or they can be bleached, and it says right here use of 10% bleach, a 1 to 10 dilution of domestic bleach is all you need. You can burn them, biosafety pickup, or use an autoclave if you have one. So just depending on what you're doing, depends on what you need. But bleach is the easiest way to go. Double bag it and you're good to go. Any proposed changes, if they want to change anything, they've got to come back to you and talk to you again, because they may change something that shifts them from a BSL-1 to a BSL-2. So you don't want that to happen without your knowledge and making sure that they're in the right place. Because I know last year, this is one thing I had to call a 6th grader for, is they had grown unknown organisms in their home, which is what we're moving into, and it disqualified her because there were a lot of things not signed off and you can't do that. So with your unknown microorganisms, so these are the things that they're going to want to get off the drinking fountain at school and off toilet seats and all that kind of stuff. So any unknown organisms, they typically involve those collections of culturing microorganisms from the environment, so household surfaces, skin, soil, anything like that. That's what is included in this group. So with unknown organisms, they can be treated as a BSL-1 under the following conditions. This is the big part. The organism is cultured in a plastic petri dish and sealed. So as long as they put it in there and seal it and never open it again, they're fine. But it's got to be at the high school, okay, or at whichever lab they're going to be at. The experiment involves only procedures in which the petri dish remains sealed. So there you go again. It's got to be sealed the whole time. It's got to be in their research plan that it's remaining sealed. So they can obviously still count presence of colonies if they're going to do something like that. But the sealed petri dish has to be disposed of under the supervision of whoever's supervising them. So with that bleach, that's the person that has to do that, and that's got to be sealed all the time. Anything with an unknown organism cannot be opened for any purpose, or if it is, it's got to be done in a BSL-2 lab because they've got the ventilation, they've got the PPE, the equipment that they can put on and make sure that they stay safe and don't get contaminated in any way. Now, I will say this rDNA technologies, that's another one. I've never worked with anybody that's done this, but it can get a little crazy too. So the rDNA technologies where organisms, plants, or animals have been genetically modified. So this is where they're genetically modifying things, require close review to assess the risk level. So they want to make sure that everything's going to be safe, again, which is, our main goal with all of these rules. So this type of study has to be done in a BSL-1 laboratory under supervision. So examples of this, they're cloning DNA in E. coli 12. Okay, so those kind of things. It's got some other examples there. The BSL-1's agents may convert to a BSL-2 agent. If that's going to happen, it has to be done in a BSL-2 facility because obviously you don't want contamination in general. Anything with a BSL-2 organism has to be in a regulated research institute. So just like I said, this one can get kind of crazy depending on what they're doing and how brave they want to be. The propagation of recombinants containing DNA coding for human, plant, or animal toxins, including viruses, is prohibited. They cannot do that at all. The genome editing studies that include alteration of germline cells, insertion of gene drives, all of that's going to have to be done in a BSL-2. Like I said, this one gets a little crazy. The introduction of or disposal of non-native genetically altered or invasive species, pathogens, toxic chemicals, any of that into the environment is prohibited. So obviously everything has to be bleached and disposed of. None of this kind of stuff can be just tossed in your regular trash can. But make sure that it is taken care of appropriately. All right, some additional rules for projects with tissues and body fluids. Now, this is one thing that we talked about on Tuesday. Somebody had asked about a deer, I believe. Somebody had asked if their father shoots a deer and they want to take a tissue sample. This is where that's at, that they actually cannot do that. When I was reading through this, I figured that out. So with this one, they may contain microorganisms, or this tissue or anything that they're taking from a vertebrate, so that deer, it may contain microorganisms and have potential causes of disease. So proper risk assessment is required. Human or non-human primate established cell lines or tissue culture collections, they have to be considered BSL 1 or 2 as indicated by the source information and treated accordingly, and we're going to get to that. The source and catalog number of the cultures has to be identified in that research plan. So if they buy these tissues from somewhere, then obviously they need the catalog number of where they got those cultures from. If tissues are obtained from an animal that was euthanized for a purpose other than the student's project, it may be considered a tissue study. The regulated research institution requires a copy of the IAC-UC certification with the name of the research institute, and it tells you the rest there that they need. Tissues obtained from agriculture or aquaculture studies require prior SRC review. If the animal was euthanized solely for the student's project, the study must be considered a vertebrate animal project and is subject to vertebrate animal rules. Usually that's a big no if they're euthanizing an animal just for their project. If they're being euthanized regardless, then they're usually okay. All right, so the collection and examination of fresh frozen tissue or bodily fluids from a non-infectious source with little likelihood of microorganisms present, they're considered a BSL-1. It has to be conducted in a BSL-1 lab and supervised by a person, scientist, designated supervisor, whichever one. And here is, it says meat or meat byproducts not obtained from food stores, restaurants, or packing houses may contain microorganisms. Because of that increased risk, that goes to a biosafety level 2. That's where if they're collecting like deer tissue from that obviously wild deer, they're going to have a BSL-2 study. They're going to have to work with an institute. So that's not something that even with the hunting permits and everything that they're going to be able to do. And number 7, we're going to get into that too. Number 6, I had to share this one with my whole team last night or yesterday when I hit this. Human breast milk of an unknown origin, which is just, I don't know where else it would come from, unless certified free of HIV and hepatitis C, which you can, I guess, get those from donors from a hospital. Hospitals do take donations. So obviously, they would know the origin at that point. In domestic unpasteurized animal milk, they are considered BSL-2. So basically, anything they buy in a grocery store, they're good to go with. The things such as this, they got to be careful. All studies involving human or wild animal blood or blood products, they have to be considered a minimum of biolevel safety 2. So again, the deer, if they choose to do that, that's going to have to be a BSL-2. And I know a lot of our students are exposed to wild hogs and deer and hunting and that kind of thing, but just because of the unknown organisms, it can be an issue. So studies involving domestic animal blood may be considered BSL-1. So obviously your horses, your dogs, cats, any of that kind of thing, that's just a BSL-1. It's just when it's unknown it gets a little scary. All right, any tissue or instruments with the potential of containing blood-borne pathogens has to be properly disposed after experimentation. So again, bleach, that usually doesn't require autoclaves. Bleach is going to kill everything. Human body fluids where the sample can be identified with a specific person obviously would need review, approval, and informed consent from that person. Any study involving the collection and examination of body fluids that may contain biological agents belonging to BSL 3 or 4 is prohibited, and we're going to get into those categories shortly to know exactly what that means by BSL 3 and BSL 4. So students can use their own body fluids as long as they're not culturing anything. They can be considered a BSL 1. They can actually conduct it at home, but they have to have an IRB review if the body fluid is serving as a measure of an effect of an experimental procedure on the researcher. So just for example, it's not going to need an IRB review if they're just collecting urine to serve as a deer repellent. And then they must receive prior SRC review and approval prior to experimentation. Studies involving embryonic human stem cells must be conducted at a research institute. It's usually pretty tough to get those anyway. I've never had a student do those, but it could definitely happen. Maybe we'll have the next Einstein. So some of the exempt studies, these may be subject to additional rules just dependent upon the design of the project, but these are some of those that they don't need that prior pre-approval. But they do all require a risk assessment, but everybody's does. Any study involving Protus or archaea, they can go. Anything using manure for composting, fuel production, or other non-culturing experiments, they're exempt. Commercially available color change coliform water test kits. This kit has to remain sealed and must properly be disposed of. Studies involving the decomposition of vertebrate organisms, so those are going to be your forensics projects. Yes, and Ms. Ryman did confirm yes, they do have the human milk banks that students could go to if they wanted to do a breast milk study. And then I actually did have one today, so this was exciting. So Ms. Ryman had one at her school, studies with microbial fuel cells. So they've got one going on out there. So that one actually is exempt, but we looked at that one today, and she's good to go. So that one will be exciting to see. I think that's my first one. So the following types of studies that are BSL-1 organisms, these are exempt as well. If they're just using baker's yeast or brewer's yeast, and most of this can be bought at the grocery store, they don't need to have that prior approval. And then studies involving your nitrogen-fixing, oil-eating, algae-eating bacteria introduced in their natural environment, those are okay. Obviously, if it's going to be cultured, it's an issue. Once they start culturing things and growing things, then it's always going to require more paperwork. So some more that are exempt, your water or soil microbes that are not concentrated in media that are conducive to microbial growth. So again, nothing that can culture. Studies of mold growth on food items. And Donna, I think we had one of these at your school today as well that somebody was wanting. This is what I was looking for. Mold growth on food items if the experiment is terminated at the first evidence of mold. So they can grow it, but they gotta toss it. So they're gonna have to make sure that that's in their procedures correctly. Studies of slime molds and edible mushrooms, and then E. coli, K12 is usually pretty good to go, and they just buy that from Carolina Biological most of the time. So exempt tissues, so these are ones that they don't need that pre-approval for. Any kind of plant tissue unless it's toxic or hazardous. Obviously, we don't want them eating toxic plants. Plant and non-primate established cell lines and tissue, those culture collections, they're usually obtained from a collection source. Fresh or frozen meat, I think we hit on this earlier. Anything from a food store, restaurant, packing house, and it's been pasteurized, so they're usually pretty good. Hair, hooves, nails, and feathers, they're okay. Teeth. Now, guys, I will say this. teeth that have been sterilized to kill any bloodborne pathogen that may be present, if they go to a dentist and get teeth from them, the dentist has to provide a letter saying that those teeth have been sterilized. So we had a tooth study last year, and she had to have that in order to get through the fire team at state. So that's just something to be aware of if you have anyone doing teeth. So your fossilized tissue or archaeological specimens or those prepared fixed tissue slides, which we all know those are good to go. All right, moving on to what the heck these PHBAs actually are and some of their levels. So this is just a risk assessment if you run into a PHBA and trying to figure out if it's a bio level safety 1 or 2. This is where you go to kind of help and think about it. As far as this whole assessment defines that potential level of harm, injury to plants, animals, or your students when working with these specific agents. So this assessment kind of determines that lab facility, equipment, training, the supervision that you're going to need. So the first thing you got to think is the risk group, the initial assignment of that microorganism to that BSL level, and then those unknown organisms. Are they using fresh tissues? Rely on the expertise of some of your supervising adults. Determine that level of biological containment they may need. Look at that experience and expertise level. So a biosafety level for study based on risk group or biological agent. So make sure prior to experimentation, if the study is conducted at a non-regulated site, if the study was conducted at a regulated research institute, so think about where they're doing it. Was it conducted at the regulated research institute, but that institute did not require a review of this type of study? So just these are things to think about when you get hit with these types of projects. So with your BSL 1, this risk group usually contains biological agents that pose a low risk to personal environment. They're highly unlikely to cause any disease in a healthy laboratory with workers, animals, or humans, or plants. So they give you some examples there. And then if you look over to the actual level of containment, it defines BSL-1 and what it looks like. And it even says, normally found in water testing labs, high schools, colleges teaching introductory micro. Work is done on an open bench, biosafety hood. So it just gives you decontamination can be achieved by treating with chemical disinfectants. So really simple, just what does that BSL-1 lab look like? And then with the BSL-2, that group of agents pose that moderate risk. So there is a risk of spreading whatever they're working with to someone else or some animal or anything like that. It rarely would cause an infection, but we do know at least there are effective treatment measures and preventative measures that are available if something was to go wrong in the lab and they were to be exposed. So here are some of they've got some examples there of the organisms that need to be handled in a BSL-2. And then that BSL-2 lab, you're looking at moderate risk to humans. So access to the laboratory is usually restricted. So you have to go through, it's almost kind of like some of the ones you see on TV where they got to walk into the safety areas maybe. The autoclaves always there, lab coats, gloves, eye protection, face shields. So those types of areas, that's what a BSL-2 lab is going to look like if you need that picture in your head. Doing a lot of tough stuff this week. All right, so there's some fun stuff in here too with your hazardous chemicals, activities, or device rules. So these do include substances and devices that are regulated by local, state, country, or international law. So you got to make sure you're at least semi-aware, or the person that the student is working with is aware. Hazardous activities are those that involve a level of risk above and beyond what's encountered in the student's everyday life. So that's what you always got to turn to, is that everyday life statement and kind of assess that situation. So all of these projects, they have to just be described in the research plan, the risk assessment process, supervision, safety, methods of disposal. They do all require direct supervision. The student must acquire and use regulated substances in accordance with all of the laws that are laid down for those things. For all chemicals, devices, or activities or acquiring federal or state permits, the student or supervisor must obtain that permit prior to the onset of experimentation. So the student research, in essence, must minimize the impact of an experiment on the environment as well. So they don't want to go do a, controlled burn in a pine forest if, that's not necessarily something that they need to be doing anyway, or putting chemicals in the environment that don't need to be there. All right, so your DEA controlled substances, this regulates the chemicals that can be diverted from their intended use to make illegal drugs. So that's going to be your Sudafed. Okay, so this is usually the things, or even cold medicine, that they ask for your ID at Walmart. So all studies using the DEA controlled substances must be supervised by that qualified scientist who is licensed for the use of that controlled substance. So obviously going to depend on what it is. All studies using the DEA Schedule 1 substances, including marijuana, because I think I have somebody asking about somebody asked at the meeting about the gel or the liquid, must have the research protocol approved by DEA before research begins. So the student would be responsible for that. With any kind of prescription drug, your students are prohibited from administering prescription drugs to human participants. That's not something they can do. A veterinarian must supervise student administration of any prescription drugs to vertebrate animals. So obviously, if the student can't do it, a doctor could, but it would be an observation project at that point. And we hit this one the other day, fermentation studies in which minute quantities of ethyl alcohol are produced are permitted, but the designated supervisor is responsible for taking that away using appropriate disposal of the alcohol or tobacco used in the study. So as long as the parents or supervisor or whoever's there that's obviously planning on purchasing that for them and allowing them to work on that in their home, they need to be there. Production of wine or beer by adults is allowed in the home, but they have to have those regulations and understand that. Students are allowed to design and conduct that research project, but it has to be under that direct supervision, and it has to be legal. Students are prohibited from conducting experiments where the consumable ethyl alcohol is produced by distillation. However, the students are allowed to distill alcohol for fuel or other non-consumable products. So to do so, that work has to be conducted at school or a regulated research institute. And somebody always wants to do something with guns. So just so you're aware, a firearm is defined as a small arms weapon from which a projectile is fired by gunpowder. An explosive is any chemical compound, mixture, or device, the primary purpose of which is to function by explosion. Explosives include, but are not limited to, dynamite, black powder, pellet powder, detonators, and igniters. So always remember, I think, and there is one down here too, that we're going to get to paintball guns and potato guns, because those always come up for some reason. So we do have that laid out here. So the purchase of the firearm by a minor is generally unlawful. So obviously their parents are probably hunters or something like that, and they've been through everything. I want to say at the age of 15, you do go through the gun hunter safety course. So the use of that firearm without proper safety certification is illegal. So they should make sure and have those training and certification card if they're going to be the ones shooting the firearm. So firearms and explosives, they definitely are allowable, but they got to be under the direct supervision of that supervisor and in compliance with all of our laws. A fully assembled rocket motor, reload kit, or propellant module containing more than 62.5 grams of propellant are subject to the permitting, storage, and other requirements of the explosive laws and regulations. I've never had anybody use a rocket motor or reload kit. I'm sure there will be a time. But here's the part, because a lot of kids love potato guns and paintball guns, but they are actually not considered firearms unless they are intended to be used as weapons. So depending on their study is going to depend on if that's a weapon. But we know we can't create pain, so they shouldn't be shooting at each other. But they do need to be treated as a hazardous device. If anybody has any kind of drones, number one, if they go into display and safety, they cannot have any batteries in those drones when they go into the fair. That cannot happen. They cannot fly the drone in there. And it also has to be registered with the FFA, So the Federal Aviation Administration, they have to be registered with that if they're doing drones. Radiation. So you will have students that may want to do things with x-rays. So it's all going to depend on the level of exposure and the radiation release from those sources, because those can be a health hazard to students. They may not exceed the dose limits set by the Nuclear Regulatory Commission, and it gives you the regulations there. The voltage needed in a study has to be less than 10 kilovolts, and I did have some voltage questions and concerns last year. So the study may be done at home or school, but they do need, oh, and it says SRC pre-approval is not required, but just make sure you check those volts and ask for that kind of thing when you're looking through those research plans. A study using 10 to 25 kilovolts must have a risk assessment conducted and pre-approved. Those have to be conducted, or similar studies, these types of studies, must be conducted in a metal chamber using a camera only, not direct view through the glass. So a dosimeter or radiation survey meter is required to measure radiation exposure. So all studies using more than 25 kilovolts must be conducted at an institute with a licensed radiation program. So I haven't had any radiation studies yet. Here's just in general for hazardous chemicals, hazardous devices. This is that guidance for the risk assessment. So when you have students that you know have these kind of items that or this kind of study, first thing to think of with your hazardous chemicals, think of toxicity, reactivity to other things, flammability, and corrosiveness. So think of those four things immediately, and kind of, to think about that risk assessment and what they need to do to stay safe. I know we had a glow stick one today, and we didn't know how toxic a glow stick was, so that's something that we had to look into. Your environmentally responsible chemistry. Avoid the use or production of hazardous substances during the chemical process. So you don't want to, take one thing and turn it into something that's more dangerous. So these things, whenever possible, as far as this goes, make sure they're incorporating these types of things into the research plan: the waste prevention, renewable materials, maximization of energy efficiency, those types of things. So that's highlighted just to make sure you think about it when you're looking at their risk assessment. The type and amount of exposure to a chemical must be considered, especially if they're, not wearing gloves, which hopefully they are. But they, you got to remember that some of these chemicals are definitely not good for long periods of time. Again, risk assessment forms required. So some hazardous devices, some commonly used devices: Bunsen burners, hot plates, saws, drills, may not require a documented risk assessment. I disagree with that. Assuming that the student researcher has experience working with the device. Now, I will go ahead and tell you, I had one that worked, what was it? I can't think of the name of it. It was something that burned into something else, a soldering iron. The dad we had to go back in his research plan and put in there that dad was present, and he was, but they had forgot to put it in the research plan, and they were actually going to not allow him to participate at state if he did not fix this in the research plan. So anytime they're using those saws and drills, anything, just have a adult supervisor there and make sure that it's in the plan. During the researcher's time of using the saw, the designated supervisor was supervising, just something like that. Okay, I have a question here. It says wood burning for scrimshaw. Is that where they're just writing in wood?
Scrimshaw is burning into like bone or something.
Oh.
That's where they used to take the like tusks and burn into it.
Oh, okay.
That's scrimshaw. Wood burning is just burning into wood.
Mm-hmm.
Scrimshaw is like burning into like bone and stuff.
Oh, okay. I've actually never heard of that. Sandra, is that something that they do all the time? The student, is that something that their parents do as a hobby? I guess I'm trying to put some context to it. Because if there's a risk of the student getting burnt, then they may want to have a supervisor there. Yes, they are hobbies.
Yeah, those things get wood burners get super hot. I use them myself, and yeah,
That's
you
what I
need to have
thought.
a supervisor there. Absolutely. because I mean, all you do is touch it to the wood, and it starts burning in to the wood.
Okay. Okay. I would definitely, especially if they are hobbies, if the parent, I would want to designate a supervisor there in case the student got burnt.
Hmm.
So I would just make sure and put that in their research plan. Okay, and then there's some other listed equipment there. Like I said, just make sure that they put the safety aspects there because we had some issues on several of them last year with that. So radiation, if it involves radiation beyond the normally encountered in everyday life, but this does include UV, infrared, microwave, any of that kind of thing. Just make sure you're considering it when you're reading through the research plan. Okay, this for the engineering project guide, these are just some questions. I didn't highlight these because they're a little bit different, but these are just some that you can consider, have your students consider when they're creating their projects. When it tells you in the research plan that you need to have things like aseptic technique documented and some procedures for those types of things, this is those sources of information. So over here, information for students, that's the APA. You know, that's how to cite APA. There's some other things in here that like the right here, guide for the care and use of laboratory animals or any kind of animals. That kind of stuff needs to be cited so when they discuss the care of the animals during their research project or anything like that. That's what this whole set of sources is for, is so that they have those things, the patent office, all that kind of stuff. That's what this section's for. This is where they recommend them go. So fisheries.org for anything of fish related. So I'm not gonna, just wanted to point that out that that's what that's there for. All right, so for display and safety, for your students' boards, this is the responsibility of the finalist mostly. Definitely as the adult sponsor, fair director, I'm going to be there. We have a display and safety at the fair. We have a whole team that comes in and does this. So they need to make sure they watch the sizes of their projects because we do have to measure from the floor to the top if it's a floor project. If it's on the table, they've got to meet certain depth and width. Some people just make these gigantic boards. Some people do go have them printed and they're just the pop-up signs. I've seen those. If they get real crazy and want to take that type of thing to state or have them printed up on the big, beautiful display boards, that kind of stuff is fine. Just to tell them they've got to make sure they watch those displays because you don't want them to get disqualified for something that really could have been prevented. So it has to fit within the project dimensions. And this says including table covers. Please know if you haven't been, I've got tablecloths for the tables and students do not need to use that. Sandra asked, so they put this information in their bibliography. I'm guessing that we're talking about the sources page that we were just talking about. If they use any of those resources, especially like the animal care, just that general stuff, they need to make sure they cite it in their bibliography. I will tell you, last year I was surprised at the ones that were going to state, they only had one thing cited in their bibliography. If you have anything with humans, vertebrates, anything like that, you've got to have 5. And that includes ICEF in the state. And there's 2 right there. So they really only need 3 more. But if they talk about how to care for animals, they've got to cite that in there. It's highly recommended. It makes it a lot easier to get through the fire team. Okay, so if a table is used, it does become part of the project, but so it still can't exceed those dimensions. It must be done within the confines of the finalist booth space, any demonstrations that they choose to do. Projects may not lean against, it says the back curtain, the way they're going to be set up at Big Springs is they're going to be back to back, on both sides of the table. So they need to make sure that their project doesn't have to lean up against something. So if it's going to be propped up, they need to make sure they bring whatever's propping that up with them. Donna just asked how tall are the tables at Big Springs. They're the regular height tables. trying to think about how tall they are. I know they're the
I
eight
think it's
foot long ones.
36 inches, isn't it?
That sounds right. Yeah, that
I
would be
think
about
that's
right because that's about at my waist. The tables are about at my waist, and I'm 5'1", so that would be about right. And please too remember, if they have a display and safety problem at Big Springs, that's not going to disqualify them for us, because if their project is good enough for state, then I'm going to have them fix it before we go. So we've had, I know that's happened in the past with Jackie. We didn't have any issues with that kind of thing last year, but I'm sure it's going to happen. So the front edge of the the placement of the required forms. So there are some forms that are absolutely required to be at the booth, and then some absolutely have to be on display. So these may be on the front edge of the table, the display board, or freestanding in an acrylic frame placed on the tabletop. So just depending how the student wants to do that and show that, that all depends on how they want to do it. But the forms that are required at the project, the official abstract has to be there at all times. I've had them tape it to the front. I've had them put it on their board, but that one is required to be displayed. This was a tricky one, and this got a lot of people at ICE at International. The term abstract cannot be used as a title or reference, so you can only actually have the word abstract on your board once. Period. So they've had to go in and rip them off and try to cover them up and make them look pretty. So just make sure that the word abstract is only on the board one time. Oh, so what's going to happen is our display and safety team, we use the form from state. We're actually pretty known well at International, that I learned this year. They will walk by a project and go, oh, that's from Florida, we're not worried about it, regardless of how crazy it may look, because our display and safety is so much more strict here than they are at International. And most of our display and safety team actually goes to International too, which is impressive. I guess that's not what I'm going to do with my retirement days, but if anybody is looking for something interesting and likes to travel, that would be something fun to go do. Another form that has to be at the project is if it's a continuation study. Form 7 has to be completed and vertically displayed at the booth. So that one and that abstract paper, that has to be there. The abstract must reflect only that year's work, and a reference to the past work on the display board has to be limited. So they don't really want a whole lot of information about what they did last year. Even if it's a continuation, that needs to be short, sweet, and to the point, and moving on. Now, all the forms need to be at the project. The rest of the forms need to be at the project. They don't have to be displayed. They can be, in their logbook, in a folder, anything like that, but it does still have to be there. Forms that they do not have to have at the display booth or even in the exhibit hall. Any of those completed informed consent or assent forms, they do not have to be displayed. They should not be present at the project display. That is people's personal information. That needs to be, we'll just call it, locked up at home. Now this one we've had some issues with in the past. Make sure you check your students' photographs that they are putting on their board. They cannot be offensive or inappropriate, and what they mean by that, they don't want to see any kind of surgeries. They don't want to see anything, a necropsy, dissections. None of that can be in the pictures on the board. They need to have, whatever pictures they do have, it has to have a credit line. Photograph taken by, or image taken from, graph or chart taken from. There has to be some kind of label with those pictures. That's an issue we always have, even at regionals. The students forget to put that. I will have labels there, but it's best that they just have it done so that they're not stressing when they get there about things that they're getting cited for with the display and safety team. And all those images must be properly cited. The signed photo or release form is in the notebook or the logbook. So those consent forms do need to be available. So if somebody agreed to allow them to have their photo on their board, that one needs to be available for the judges. So a sample release, I consent to the use of visual images involving my participation in this research. It's that simple. So if they have that and it's signed, they're good to go. It's just saying, you're okay with your picture being out there, like on Twitter. So some of the students Ms. Crusher asked, don't they need a redaction at the time of judging for human studies? Yes, ma'am, of that of the consent form. They do need to have a redacted copy with them to just make sure that the specifics of any person are blacked out. So some of the students want to do a like PowerPoint presentation or demonstration outside of the project board. So some examples of those, but these do require approval from the display and safety team. So if they want to do it, they're going to have to do it for them first. It'll have to be approved before they can actually do that. So some of them, they're limited to a PowerPoint, a Prezi, Keynote, YouTube, software program, simulation, or other images or graphics displayed on a computer screen or other non-print delivery method. So some of the things not allowed on display. Hang on. The students cannot have any acknowledgments, self-promotions, or external endorsements. So they cannot do a project that's endorsing their parents' company. The use of logos, so any brand names, crush, trademarks, none of that can be on the board at all. Period. They can say paper towel 1, paper towel 2. They cannot use the actual brand name. So personalized graphics or logos that is developed to indicate a commercial purpose, so creating that sales gig. Any reference to an institution or mentor that supported their research, a reference to a patent status. There are a lot of students in international that do register for patents and are working on things. That can create bias with judges, so they are not allowed to do that. Items intended for distribution. They're not supposed to be distributing anything. The only thing they can distribute is the certified copy of an abstract if they wanted to do that. So discs, CDs, flash drives, brochures, any of that kind of stuff, they cannot be there giving away. Any awards or medals for past awards in science cannot be done or cannot be there. Addresses, World Wide Web, email, social media, they don't want that there either. Active internet or email connections as part of the display cannot happen. Any changes, modifications, or additions to projects, including any attempt to uncover, replenish, or return removed language or items after the approval by the safety committee. So once that safety committee has said, yep, you got your green sticker and not a red sticker, you're good to go, they cannot do anything. They're good. They can't change anything. So this is where this gets kind of fun too, because we always have students wanting to bring things, and sometimes they're living. If they did plants, they cannot do that. So these certain things are not allowed at your projects. Any kind of living organism, glass. We had 2 projects last year, believe it or not, that had to do with cement. They cannot have that there, even if it is enclosed in something. Taxidermy, preserved vertebrates, human or animal food, human or body fluids, plant materials, any chemicals, including water. Okay, they do not need to have any kind of water on display. Let's see, hazardous substances or devices. So example, if they were doing firearms, none of that should be there. Items that may have contained or been in contact with those hazardous chemicals. Sharp items. So if they were dealing with syringes, needles, leave it at home. Anything flammable. Batteries with open top or wet cells. Drones. Now this is where they have to make sure, like I said, the propulsion or power source is removed. No batteries can be in the drones in the display or exhibit area. 3D printers, see here, unless power source is removed. So it can be there, but it can just be for show, just for looking. So anything that's inadequately insulated and is capable of producing temperatures, cannot bring it. Any apparatuses with belts, pulleys, or chains, moving parts, and any display items that are deemed distracting, so sounds, lights, odors that can distract others. Personal items or packaging, they cannot store anything underneath their table. We make exceptions for that with Big Springs, but any apparatus deemed unsafe by the SRC. And then electrical devices have to be protected. Any enclosure has to be non-combustible. Metal parts have to be grounded. Your energized wires, switches, and metal parts have to have insulation and overcurrent safety devices. And it must be inaccessible to anyone other than the finalist. So if they have something that's plugged in, it's got to be unplugged, that kind of thing. Guys, this was a big one last year, and I felt so bad for the girl. Decorative lighting or illumination, it says it's discouraged. Light bulbs are prohibited. I had a student last year that went to state. Mom and I had words about it, and I told her, I said, I would highly recommend you take the light bulbs off and take them off now, because when she gets there, if display and safety tells her to take them off, it's probably going to ruin her board's going to get ugly. Well, she's, well, we're leaving it. I said, okay. I said, I'm just letting you know, if she's disqualified, you're going to have to come pick her up. They didn't do that, but they made her take the bulbs off, and it tore up a lot of stuff on her board, and she was not happy. So it was just very upsetting. So make sure students don't put decorative lighting on their board, because they're not going to be able to keep it. The state will not allow it to happen. An insulating grommet, if at any point wire or cables is entering anything, no exposed live circuits, and here you go, over 36 volts. We had some people pushing that limit last year. It must be accessible, clearly visible, an on-off switch, okay, with a means of quickly disconnecting it if something happens. Your lasers, class 1 or 2 lasers, along with the 3A, are allowed, provided finalist avoids indiscriminate exposure to other people. So they have to be labeled by the manufacturer. They will not allow labels or lasers without labels. They will not be able to bring them in. The LEDs that consume over 1 watt will not be allowed. Handheld lasers, absolutely not. And ladies and gentlemen, I believe we have finished this entire book. Yep, we are at the forums.
All right.
Any questions, concerns? Exactly. Miss Crusher says, yay! It's a lot of information to absorb, which is why I wanted to break it up.
Well, I'm waiting to send my BSL1 paperwork in with my sheet, my IRB sheet.
That's perfectly fine.
All right.
Not a problem. Well, if nobody has any more questions or concerns, then I will let you guys go, and as soon as I can get this up and working with the link, and please feel free to share the other link that I sent. I did put it on the Big Springs website so that everybody would have access to it at all times, so you didn't have to hunt emails, but please feel free to share these with your teachers, and I hope it helps.
Well, I appreciate the time.
Absolutely. And do I need to send out like a voting button or something? Did we want to do webinars where it's like a question answer session with things you have questions about at your schools? Or I remember us talking about it Tuesday. I didn't remember if any decisions were made on that.
At my school?
Oh, no, just anybody.
Oh, yeah.
Okay. I was going to say, most people just...
I think any questions we have, we're just going to bring to the paperwork session here at Lake Weir.
Okay. Sounds like a plan to me.
So.
All right.
All righty. Thank you.
Thank you so much, and have a wonderful evening.
All righty. Bye now.
You too. Bye.
Bye.